Well, it's a great pleasure to be here back on the campus again. Even more pleasurable to be able to speak with my friends and shipmates. But what I want to do this afternoon is take you on a journey to look at changes in the ocean. And we've heard a lot about stressors and climate change and those things. But what we have at our disposal is the fact that we have a body of knowledge about the Sargassum community. It came from these three primary investigators, Jim Butler, Byron Morris, and Alan Stoner, of course, Alan being a former faculty member here. And they really put a lot of time and effort into looking at the Sargasso Sea on the motivation of a stressor, marine pollution. They really were looking at the impact of hydrocarbons from waste oil discharged from ships in the Sargasso Sea, trying to see where all that oil went. So when we think about the challenge of trying to understand the biology of the Sargassum community and understand that it's moving, it's under control of oceanographic properties that move these animals over vast spatial scales, but the clock is also ticking in terms of the seasonal patterns of distribution of these plants. So we have to think about understanding the things at a very local scale, the intimate life that happens in a large mat, which will be broken up into windrows, which will be broken up into small clumps, isolated from lots of other sargassum. A very dynamic spatial and temporal system. So we need to be able to keep that in mind when we're looking at there saying, I'm studying the Sargasso Sea. At that moment in time, at that moment in place, you have to keep in mind the context of all the other processes that might deliver an aggregation of species on that front that you have to pick up with a dip net on a particular cruise. So it's a big challenge to understand all those patterns of time and space that are going on there. So one of the first studies to really give us this concept was by John Ryder, then a member of the Bermuda Biological Station, as it was called back in the day. And he helped us understand how the prevailing current systems create this gyre system to give us this concept that things are moving around. They spend time at low latitude, they move east to west, north and south throughout the year. And so when we want to look at the question of where the Sargassum is and who might be associated with it, we really have to understand that we're looking at changes over time and space that are really important to define these patterns of distribution. And if the plants are moving and the environment's changing in terms of water temperature, day length, nutrient availability, they're going to have an impact on the species that are associated with the Sargassum, whether they're living on it or whether they're moving in to use that Sargassum as a habitat for some period of time. So we go about trying to find out how much Sargassum out there with our favorite tool, the Newson net, which we drag with various vessels around. This is Morris's Newson net. This is the SEA boat. We've been doing this for, in a fairly quantitative sense, since the early 70s up to about 1976 by Butler and Morris. Alan Stoner came along to work here with SEA from 77 to 81. And then things sort of dissipated. Interest in the Sargassum community disappeared, although SEA has maintained a time series over 40 years in terms of the abundance of Sargassum, which Amy really consolidated in a report with the Sargasso Sea Alliance. So we know a lot about the plant, but we haven't had quite as much attention on the biological associates of Sargassum until relatively recently. We look at tows, they're very useful. They can have biases in terms of whether you're going with windrows or cross windrows. The ship goes on its course, the wind and the currents dictate whether the Sargassum is going to be in a particular juxtaposition. But in aggregate, we get a very comprehensive picture for the western part of the Sargasso Sea of where Sargassum has been. And so you can look back in time. This is some maps created to show historical data from Parr and his cruises. And Bermuda is always in the center of things because if you're in a ship, you got to go to shore at some point to get food and water to leave your crew. So we have some information about this part of the Sargasso Sea because that's where the vessels are transiting, because the port of call Bermuda makes it relevant to be in that part of the ocean. Alan's effort with SEA was to really expand the map of where Sargassum was occurring and to begin to understand the spatial patterns of distribution of the plant. But here's the data set that Amy has pulled from SEA records to look at the patterns of distribution and really revealing the fact that there are seasonal changes in terms of the abundance of Sargassum from really great predominance at lower latitudes in the spring, and that's what you can see here, and then you're seeing the preponderance of the Sargassum in the northern latitudes in the fall period. But a bigger story comes out of that from looking at the types of plants and the diversity of Sargassum is determining that Sargassum natans is much more distributed towards the northern latitudes. The Sargassum fluid is much more to the south. And that wasn't apparent to early biologists. They didn't spend enough time to see. They didn't have the benefit of 40 years of aggregation to tell you there is a spatial pattern in the distribution of these plants. that's a really big story to have at our disposal today. So what we think about, and we've seen some of the posters today and talked a little bit about some of the really distinctive inhabitants of Sargassum, looking at the several hundred species that are there either permanently, intimately associated, living, growing as epiphytes on the Sargassum plant itself, versus animals that are there and mobile, moving around, versus other species that are transient, they're arriving. There's the juvenile sea turtles that are there for 4, 5, 6, 7, 8 years of their early life, using the Sargassum as a habitat, using it as a place to find food resources. Juvenile fishes coming into the Sargassum community, use it for a period of time. So we have a pretty good idea of the pattern of abundance and species diversity in there, but the questions are really what's been happening over time. What we also have gained from these early studies is looking at the relationships in terms of the consumers within the Sargassum community. This is the cartoon of a food web that we know from the early studies by Butler and Stoner. And we look at who's living in there. We have the epiphytes, things like the hydroids, looking at plants, looking at the fact that there are primary consumers, a number of different types of amphipods here. We have our shrimps that are secondary consumers feeding on these amphipods. And at the top, we have our tertiary consumers. And this particular cartoon they've given us is looking at the presence of crabs, both the Bondis minudus and Fortunus as the top predators, and off to the side the two resident fishes that we have in here. But this is an incomplete picture. It's a very good study of what you can get when you use a dip net to go and haul it out, put it on the deck, take it apart and see who do we have here. But what it misses completely are all the transient species, things you can't capture easily in a Noosa net or with a dip net. Yet we know they're there. We've seen pictures of clouds of fishes underneath the Sargassum community. We've seen pictures of the turtles. You can't pick them up with a Noosa net or with a dip net. So they're absent from this construct of how this community is assembled and what the nature of the interactions are. But it's still fairly robust. It's still a pretty good picture of who's doing what in the Sargassum community, because Butler and Morse invest a lot of time in seeing where do they eat, where do they poop out, what's in their feces that indicates what their feeding history might be. So it's a very useful thing, but we have to recognize that as good as it is, it's not a complete story. So my interest has sort of been relatively recent, although as a younger scientist, when I was a research assistant advisor, I spent a lot of time on the research vessel out at sea doing biological and chemical oceanography, and the Sargassum was always around there, and I knew what Morris and Butler were up to, and I was fascinated with what they learned, but I went off to become a coral reef ecologist and do other things. But now at this point in my career, it's been great to be able to become affiliated with SCA and begin to think about the Sargassum community. And having returned to Bermuda after some time in the States teaching, Bermuda was in this point of, well, let's do something about the Sargasso Sea. The whole impetus to want to do more required that at least one Bermudian say, I'd be interested to go out there and find out what's living out there. Because no one had been studying the Sargasso community for close to 20 years. So building on the success of Morris and Butler, building on the work that Alan Stoner and Holly Greening have done here, we see very little other published information. There's a very small study published by Christy Huppert at the Bermuda Aquarium, collecting data just over 2 seasons in 2011-2012. I began some preliminary samples in 2012, and have been working more assiduously to collect more samples more recently to look at who's living in that ocean. Is that list of species in terms of the patterns of abundance, changes over time, spatially or temporally, the same as what we know from the studies in the 70s and 80s. But that's not an easy challenge. Remember, time and space are big factors. I'm stuck on one little rock in the middle of the ocean. I get to go to sea from time to time to go other places within the Sargasso community. So one of the first things you have to understand is, are you doing enough sampling? Or is your sampling representative? Do you have any biases? That's always a big challenge in ecology, having a robust data set that you can rely on in terms of the data that is returned from that. So one of the first things to understand is what are the known parameters for the distribution of organisms with respect to the Sargassum community. And Stoner and Greening really show something very significant, is that there are very significant spatial differences within Sargassum communities. While we have a model of who might be living there, it's not homogeneous across the Sargasso Sea, at least in the western part where we spent most of our time. So when we know it's not the same from place to place, that means that that model, who eats who and who's doing what to regulate populations, predators and prey, have interactions over time that can create balance or create imbalance. if that assemblage is not going to be uniform across time, then that simple model of who's doing what in the Sargasso Sea is not going to be applicable in all places at all times. And that has bearings for the successive species to flourish or to perhaps be diminished. So as an example, let's have a look at this. This is a list of species that are very common in terms of the top two. The top one, the Trudis fuciforme, everybody's favorite Sargassum shrimp. And all I want to highlight is, in their study, is they looked at stations along the Gulf Stream, the western part, province of Quebec, and the Sargasso Sea. Look at something like the Trudis, fairly common. But look at the next one, the Diopa, the small sail. Very rare in the Sargassum, hugely abundant in the Sargasso Sea. You look at Planius minutus, absent along the Gulf Stream, very abundant in the Sargassum community. So very clear evidence that the Sargassum communities are not uniform. One thing that's always challenging is when you're out there looking is, you have a lot of Sargassum around, or is it fairly sparse? Is the amount of Sargassum you're collecting as your sample adequately representative? And their study looked at collecting relatively small amounts of Sargassum. You can read the weights up there, medium-sized to large amounts of Sargassum. You look across these three different taxa, you can find within a particular region, whether you took a small, medium, or large sample, it was reasonably represented. There wasn't any bias with having a very small amount of Sargassum that wouldn't have anything in it, therefore isn't giving you much information about who's living in it. And so for these three taxa I've highlighted here, you can find that the patterns are fairly consistent. So it's very useful to know that if I'm going out sampling, I don't have to be overly concerned that I'm not sampling enough. I can get a reasonable amount of information returned from the sampling to be able to do. So I've had the luxury of using a boat called the Sea Dragon, sort of a sailing research vessel for hire. I've used it for the last couple of years, and I was out on it last weekend. I'm mostly working south of Bermuda to try to get away from the island. The island has an impact on what happens to what's around it. This is a long-term study site that Byron and Morris used back in the 70s. I really focused on collecting Sargassum. We did other work to try to look at plastic and marine debris, and we also were able to collect organisms working with Dan in 2013 to archive known taxa within the ocean. So here's what we begin to understand here. This is a comparison from just a data set we collected in June of 2013, and I picked a year, picked a month from back in 1973 and looked at the community composition of a couple of different important taxa. And you can look at some differences here. Fairly large area of ours here, but here's our favorite shrimp, the Trudis, off the charts in terms of the abundance of this shrimp compared to that particular month 4 years ago. A fairly irrelevant comparison, but there's a big difference here. I'll show you that this can be magnified. Look at this little Planius minutus, small Sargassum. There are a few of them there compared to that particular month 4 years ago. Lots and lots of little crabs. It's easy to find differences. Biology variables in time and space varies across time and space all the time. So what I tried to do is dig a little bit deeper in there, and I went and plotted the individual samples of all of Soner and Morris's data sets, all of the work that has been collected were contemporary data, which is my data, and also Chrissy Hubbard's data. So we're looking at the shrimp over here, we're looking at the crab over here. If you look at normalized abundance, normalizing the number of individuals per kilogram or per liter of displacement volume, because they're pretty much one-to-one for this plant, you can look at the historic data, about 260-odd samples, and you can see abundance up there really gets up to 600 or 700. Over here, Chrissy had 900 shrimp. It's a lot of shrimp to camp, right, guys? My data's up there as well. There's not much down here in the zeros. Look at these ones over here. Lots of zeros down here. No zeros over here. Now, there's a big difference in 200 samples versus 20 or 40 samples. I have more data to continue to plot. I don't have the data for the last two cruises to add here. But it's engaging to look at fairly significant difference in the abundance of a particular animal in this community compared to what we had historically. So that's what I'm after, trying to get a sense of, is this community composed the same way, the same number of species, and are there patterns of abundance similar or different to the were in the past? If you're thinking about a little shrimp, it looks like a tasty little meal, something a fish that might enter the Sargassum might look at. Or if there are fewer crabs available in here, who, from that cartoon of feeding relationships, if their populations are depressed over time, that in a trophic cascade of the top-down predators that Ryan talked about before, I think, then you would expect to see an increase in a particular shrimp if their predators are in less abundance than they were in the past. Other things that come out of looking at these communities was touched on earlier in terms of the calcifying species, the bryozoans, spirorbid shrimps, and barnacles that are shown down here. They're colonized as epiphytes here. They're a bit more problematic to quantify because of their patterns of growth. But how are they going to persist going forward in time with calcium within the acidic ocean? I think I'm about to run out of time, so I'm just going to conclude there. I was going to talk a little bit about marine plastics. That's the other big change we've seen in the Sargasso Sea, but I think I'm going to probably have to leave a little bit more time for chit-chat.
Please don't cut off for time. We want to hear what you have to say.
Okay. I just need a little love from the audience.
Yes,
So, when Morris and Butler and Stoner were working there, the big issue was pelagic tar. There's so much volume of oil leaking out of ships, either through accidents or vessel operations. It was a problematic issue and just completely polluted our coastline of Bermuda. So it's a really important study to look at where that tar went. We fixed that problem through MARPOL. Vessel discharges of oil precipitously declined over time, and that impact of the ocean is greatly diminished. But we know, we heard from our students today, how significant this plastic issue. So on the Sea Dragon, we started to look offshore. And this is my colleague, Caroline Box. She's been across the Pacific, North Pacific twice, across the South Pacific once, and across the South Atlantic once. And when she looked at the first sample of this Newson trawl, that was her characterization. It's a long question. So for someone who's spent that much time in the ocean looking at plastic, her first response when entering the Sargasso Sea was that it's more than she was familiar with. That was fairly disturbing. So thanks to her and her mentor, Marcus Erickson, we spent quite a bit of time looking at plastic in and around Bermuda. We have other colleagues, Dr. Bill Cooper and Brian Monte-Leon at EZNCW, also working in surface plastic at our oceanographic station around Bermuda. And we've seen this diagram before, this really monumentally significant graphic about plastic in the Sargasso Sea. But this is what it really looks like. This is a manta trawl, 2 nautical miles, 57 pieces, and that 57 pieces translate to about 25,000 per square hectare. That's what it looks like. It's small, it's well distributed, not very easy to see, but easy to visualize. But it's out there in great preponderance. So the impact of these small particles to particle feeding fishes, whether on the surface, or whether, as we discovered recently, many of these small particles are not as buoyant as we think. They do get mixed down below the ocean surface, is down to 4, 8, 10, 15 meters sometimes. They can be intercepted by vertically migrating fishes at night. So the other thing we've been trying to do is look at what comes up on our coastline, and looking at conducting beach surveys, a number of different beaches around Bermuda, to see how much there is. Bermuda's have been kind of immune to it until we started looking at it. Stuff's on our beaches, we pick it up and put it in the trash can and tidy it up and make it look nice for our visitors. But the insidious delivery of this plastic became overwhelming when you begin to look at it in a more systematic, scientific way. So you can get really extreme quantities. Here's 248 grams of plastic on a 25 meter by 1 meter transect. There's a little bit less. What we've done with our 6 beaches since 2010, I showed you a picture of the very first sample there, is look at the quantity of delivery that happens at every high tide in Bermuda by going to our study site and making the surveys sometime after the last high tide. All the floating plastic gets delivered by the last wave, and the next wave isn't as high, and the next wave after that, as the tide declines, leaves this residue, this bathtub ring, along the coastline of plastic. This is a log scale. So 0.01 grams per meter squared to the tide line, 0.1, 1, 10, 100. Look at this cluster of data between 0.1 and 10 grams. That's how much appears to be being delivered to our coastline with every tide. Now, not every day was it high. Over the 60 or 70 odd times we've done this. But we've never had a 0 day. There's always something there. It's really instructive to take young Bermudians, the students, or adult groups who are interested about the ocean, and make them go along that tide line, pick up piece after piece after piece, and then they see how much plastic's floating out there. Because when you go to the beach, you see one piece of plastic, you pick it up from the trash can. You don't really see the real story, which is this fairly constant input of plastic to our coastline. But of course, it's not always on our coastline, it's in the ocean. And so Bermuda's a very good place as a sentinel to be able to look and study the impact and the potential impact of this plastic around Bermuda. So we want to continue to try to expand our understanding of the changes in the Sargassum community. We have the luxury of being able to measure it against what we have seen in the 70s. I have great concerns about the calcifying species. They play a really critical role of taking plankton nutrients and remineralizing those nutrients and making them available through that sargassum. If they disappear, I greatly worry that the plant's not going to be as productive without that loop into the plankton community. And plastic marine debris is something that we have to worry about at a global level. Thank you very much. Sorry to run on.
Do we have any questions for Robbie? Everybody?
In 2007, we were on the RH Brown, and we put mock messes down 5,000 meters, within 25 years of the bottom of the Sargasso Sea. And we had a shipboard full of crazy people, because we brought up new species that nobody had ever seen before, and they were alive. There's a lot of stuff down there really deep that no one thinks about. And I know it's hard to get to, but the diversity that you talked about, you know, we've got so many new species that nobody's ever seen, undescribed species, and they're just too deep to get to. So I don't know.
And yet we know a great preponderance of some of them, or many of them, are capable of vertical migration, or enough that the ones that are migrating to very surface waters, they're going to move down 100 meters, 1,000 meters, and they'll come up. So the linkages between the surface waters, where all the plastic and all the sargassum is well understood, but very difficult to quantify. We certainly need to remind people that our ocean is not, you know, a 2-meter flat surface. There's so much life in the deep ocean that is connected to it.
And what we're doing with the surface waters are damaging the
The sargassum is a very significant carbon flux to the deep ocean. And there was a recent paper where they took baited the sargassum and baited mackerel, and they put them on the bottom of the ocean with cameras, and they watched them. You know, everybody ate the sargassum, everybody ate the mackerel. It's not like they turned up their nose on the sargassum that fell to the bottom of the ocean. So it's a difficult thing to do until you can visualize it. So you can show a fish that's, you know, lives in the deep ocean, it's got plastic in its gut. People aren't going to really get that, and we need to know whether that's happening or not. But most likely it probably is. Yep.
Since the Stoner and Green papers and the Butler papers, have there been any new species that have been found, like, really recently that like weren't identified in any of those?
I have to tell you, we collected samples on the weekend, and I found a nudibranch in there that doesn't look like the nudibranchs. When you look at the species list, there's a lot of nudibranchs that I haven't seen yet, and maybe one of those. But you know, that's the interest to look in there, and whether we may end up finding invasive species that have colonized it. That since no one looked at it, you don't know if they're there or not. So that's what I think is the interesting thing, is to begin to take apart that community and look for taxa and spend the time to try to identify them, which is a challenge because they're dead and they're preserved, and we try to keep them in ethanol so that we can do some molecular stuff to try to